Welcome to Everyday Science! Now, we're going to tackle about the history of DNA. The history of DNA started as early as 5000 BC when humans, the practice of selective breeding to produce crops, robots, and livestock. There have been many instances throughout the history where notable discoveries have been made about the DNA and inheritance that these have founded the foundation that we know and continue to advance today. Now, here are the scientists who contribute to the discovery of DNA. First, Gregor Mendel, the father of genetics, was the first person shed light to the way which characteristics are passed down to generation. Mendel coined the terms recessive and dominant. In relation to traits, in 1866, Mendel's paper published described that action of invisible factors in providing for visible traits predictable were genes. The next one is Friedrich Miescher and Richard Altman. In 1869, Swiss physician Miescher discovered nuclein and also discovered DNA or the deoxyribonucleic acid. In 1889, his pupil Richard Altman renamed nuclein to nucleic acid. The third one, Erwin Chargaff and Chargaff's rule. In 1940s, Chargaff found that the base composition, adenine, guanine, thymine, and cytosine different between species and that patterns between them were invariable. The quantity of adenine was equal to thymine, the same as the ratio of 1:1 seen on cytosine and guanine. The discovery called it Chargaff's rule. The last one who finalized the DNA model was James Watson and Francis Crick. Watson and Crick made a model of DNA structure approximately 2 years in the making. Their model was that of a double helix that consisted equally spaced pairs of bases connecting two strands. And that's the history of DNA structure. Next one is DNA structures and its function.
After we tackle about the history of the DNA, right now, we're gonna talk about the structure and the function of the DNA.
DNA, a polymer of deoxyribonucleotides. DNA carries the genetic information. The three components of nucleotides are sugar, phosphate, and base. These are the sugar and phosphate wraps around the periphery. Bases occupy the forming complementary.
The four nitrogenous bases are A. A stands for adenine. Adenine is discovered by Albert Schweitzer in 1885 by extracting the pancreas of an ox. And also, adenine is from purine bases in nucleic acid. T stands for thymine. Thymine is discovered by Albert Schweitzer and Albert Newman in 1893 by extracting the thymus gland of fowls. Thymine is also part of pyrimidine group. C stands for cytosine. Cytosine is discovered a year later by the same duo, Albert Newman and Albert Schweitzer, by isolating calm thymus tissue. Cyto pertains cells in G. Cytosine is also belong to pyrimidine derivative. G stands for guanine. Guanine is belong to purine group and discovered by Ludwig Udo Unger, a German chemist.
And did you know that the bond between ATCG are called hydrogen bond? But what is hydrogen bond? Hydrogen bond is a weak chemical bond that occurs between hydrogen atoms and electronegative atoms like oxygen, nitrogen, and chlorine. And the reason that they are always together because of the fact that Chargaff analyzed the DNA of different species, determining its composition of A, T, C, and G bases. A, T, C, G were not found in equal quantities because CG required triple hydrogen bond and
A and T only have a capacity of double bond. That's all for structure and its own function.
Now we will proceed to the importance of DNA.
After we talk about the DNA structure and its own function, now let's proceed to the importance of DNA. So, let me ask you, how is the DNA related to protein? DNA carries the code for protein, however, the actual protein differs a lot from the code present on the DNA. So, what's the notable discoveries beyond the DNA? Robert Hawley, Karl Corona, and Marshall Nirenberg, who won the Nobel Prize for their work deciphering how the DNA established the dogma information transfer from DNA to RNA to protein. In 1977, Frederick Sanger, Alan Maxam, and Walter Gilbert developed the DNA sequence. So what's the importance of DNA? Deoxyribonucleic acid, or also known as DNA, is a blueprint of life. DNA is vital for all living things, even plants. And it is important for inheritance, coding for proteins, and genetic introduction, guide for life and its processes. Also, DNA holds the structure for an organism's development, reproduction, and ultimately death. So now, let's proceed for the overall content of our new episode.
So this is the overall content of our DNA or the new episode of Everyday Science. So this DNA structure was made from recycled materials. So as you can see, there is a stanchion, there is a cardboard, there is a PVC pipe, and the base for a piece of wood for the base, a straw for the bonds, and the ribbon for to wrap around in the helix, in the DNA. So as you can see, the tansin represents the phosphate and sugar which wrap around the DNA. The cardboard for adenine and thymine and guanine and cytosine. The straw is used for the half bond, which is the double bond and triple bond. The PVC pipe is the skeleton of our DNA. You can see this DNA was made from recycled material. That things that we use to form this structure was thing that we can find in our home. So DNA is the blueprint of our life. So DNA consists all the genetic information, all the genetic codes that we need is carried by DNA.